Good afternoon, everyone, and welcome to the Office of the Chief Scientist Science Seminar Series. My name is Andrew Paul, and I'm a science advisor with the Office of the Chief Scientist, and am your host for this year's seminar series. On the behalf of the entire Office of the Chief Scientist team, welcome to today's event. I want to start the meeting by respectfully acknowledging that we are broadcasting throughout Alberta, which includes the traditional gathering places for peoples of Treaties 4, 6, 7, 8, and 10, and the Métis. Wherever you find yourself today, whether that's in Alberta or beyond, I encourage you to recognize and acknowledge the relationship that First Nations, Inuit, Métis have with the land upon which we all live. In this tradition of gathering, these science seminars provide an opportunity for us to get together to exchange knowledge on interesting science happening across Alberta in an open learning and sharing environment. The intent is that through our dialogue today, we're able to improve our awareness and understanding, but also to put that knowledge into practice to support evidence-informed decision-making wherever that might be called for. So to help maintain our call quality today, you'll notice that your video and audio has been disabled, but please enter any questions or comments that you have on today's presentation into the chat window at any time during the presentation. And then at the end of the presentation, I will read those out to the presenter to respond to those questions, and I'll do that in the order that those questions or comments are put into the chat window. So with that, I would like to introduce today's speaker, which is Dr. Holger Wille, who will be presenting on developing a vaccine for chronic wasting disease. Holger studied biology at the University of Hamburg in Germany and completed his PhD at the Max Planck Unit for Structural Molecular Biology, also in Hamburg. He later moved to San Francisco, California for his postdoctoral training, which he completed at the University of California, San Francisco, in the laboratory of Stanley Prusiner, the discoverer of the prion principle and subsequent recipient of the Nobel Prize. Following his postdoc work in San Francisco, Holger stayed at University of California, San Francisco with Stanley Prusiner, first as an adjunct assistant professor and then as an adjunct associate professor, continuing his studies on the structure of the infectious prion protein. In 2012, Holger was recruited to the University of Alberta to join the Center for Prions and Protein Folding Diseases, which enabled him to start his own laboratory. He became the interim director of the Prion Center at the University of Alberta in 2022, and then director in 2023. In recent years, his laboratory focused on developing structure-based vaccines for chronic wasting disease, as well as Alzheimer's and Parkinson's disease. And with that, I will turn things over to you, Dr. Wille.
All right. Thank you for the introduction, Andrew. And thank you all for the invitation to present at this seminar series. As you can see from this first slide, I'm combining structure biology, which is shown here on the left-hand side, the structure as it is determined by cryo-electron microscopy of the infectious prion protein, in this case, for chronic wasting disease, and of course, on the right-hand side, one of the species that's afflicted by the disease. I should also start out with the territorial acknowledgements that the University of Alberta is located on territory 6 and 7 and 8 lands and the homeland of the Métis, and the University of Alberta respects the sovereignty, lands, histories, language and knowledge systems and cultures of all First Nations and Métis people. I also have to disclose that part of what I'm presenting today was covered by a patent that was issued last year. I'm also a co-founder of a startup company that wants to commercialize some of it. However, this is mostly for the vaccine basis going towards Alzheimer's and Parkinson's disease, not so much for the prion disease vaccines that I'm going to talk about today. Now, my talk is structured like this. I'll give you an introduction of what are prions and prion biology. Then I'll show you then how we translated this knowledge into structure-based prion vaccines. I'll show you some data in a transgenic mouse model of a genetic prion disease named Gerstmann-Sträussler-Scheinker disease, quite a mouthful, what we have there. And then we translated this one and brought it into testing this one in white-tailed deer and Rocky Mountain elk. Show you some of the data there of work that is ongoing. Then we also switched over trying to make it a mucosal vaccine that can be given mucosally, in this case intranasally, and that is being tested, and we just started that work, and also briefly mentioned some future work that is in the plans right now. Now, what are prions? So prions are an unusual infectious particle in that they are protein only, which is then also involved in giving the name prions. Prion diseases, you may have heard about several of them, are found in animals, mad cow disease being a very prominent one, but there's a whole range of other animal diseases. And of course, we have chronic wasting disease, the main topic of today's lecture here, which is spreading uncontrollably in North America and some other places. We also have prion diseases in humans, and there's a variety of diseases there. I'm only going to mention Gerstmann-Sträussler-Scheinker syndrome because of the mouse model that we use there for the testing of our vaccine. So these are the prions in mammals. However, prions were discovered also to exist in fungi, in yeast and filamentous fungi. However, these proteins have nothing to do, nothing in similarity with the mammalian prions, except that the proteins as well. They don't even cause the disease in these organisms. They have different functions there. The reasons why I'm mentioning is I want to highlight this HETS protein, which is coming back to it because it is a very nice, useful tool for us. And it is a fungal prion, however, it is not disease causing, it is completely benign. Now, the human prion diseases, the common one are Creutzfeldt-Jakob disease, which is a sporadic disease, meaning it's spontaneous conversion potentially or somatic mutation. We know the inherited forms of this disease, including the mouseful one that I used earlier, Gerstmann-Sträussler-Scheinker syndrome, and I come back to this one, where in this case a point mutation in the prion protein gene gives people the disease. There are other mutations, deletion, insertions that are also giving you inherited forms. Now, the prion diseases also come as infectious diseases, and there's a more colorful ones. The one that is also of prominence here is called variant Creutzfeldt-Jakob disease, which we believe to be mad cow disease when it was transmitted into humans. Now, mad cow disease obviously has its own side of fame. First discovered in '87, transmitted through feeding meat and bone meal back to cattle, so basically industrial cannibalism. We got lucky in the sense that it did not contaminate the pastures and pens, and with stopping the industrial cannibalism, the classical BSE, which is the common one, is essentially extinct, and that is obviously a very good thing that this disease is no longer a big problem. We also discovered that, similar as in humans, this disease can show up sporadically, meaning in very old cows. We sometimes see the disease, and we believe it is the same sporadic form of a disease that pops up by itself in these old cows as we saw with the sporadic disease in humans. And we have also found genetic forms of prion diseases in cows where they had a mutation or an insertion or other alterations of the prion protein gene causing a disease there. But then again, these are very rare and not a big problem. Now, as most of you will be aware, mad cow disease obviously was a great problem in Britain, particularly in the United Kingdom, where a great number of animals got infected, spreading. And the numbers you see here on this slide are only the cows that were clinically diagnosed. There were many more animals that were incubating the disease that never got to the old enough to develop the disease properly. Now, mad cow disease was also found outside of Europe. And of course, pointing out there was 21 cows that were found in Canada and predominantly here in the province of Alberta. Now, these 21 cows caused over $6 billion of damage in the province of Alberta alone. And given the damage that was caused, this one was then the reason why the Center for Prions and Protein Folding Diseases was founded here at the University of Alberta. And so there's a direct correlation between these 21 cows and the fact that I have a faculty position at the University of Alberta. This is a direct cause and effect. Now, the disease I'm going to talk about today is chronic wasting disease, which started out in Wyoming and Colorado area. And in the gray areas, you see where the disease is spreading in the wild animals. Red dots shows deer and elk farms that have an active outbreak, and when they turn yellow, all the animals have been euthanized and the active farm is no longer active. As you can see over the years, the disease is spreading throughout North America, both in the US and as in Canada. This is now the most recent map that is available. This is from July 2025. And you see the extent of the disease both in Alberta and other Canadian provinces and down south. And it has spread, and we have no measure of controlling it at the time being. This map is also the last map that will be available because the USGS has unfortunately laid off the scientist who was responsible for this map, so we will not have updates how the disease is going further. Now in Alberta, we are relying, of course, on the map that is being put forward from the Ministry of Environment Protected Areas, which shows us the extent of the disease in Alberta in the different hunting areas. And as you see, this is predominantly a disease in the south, but if you were to go in the time lapse, which I didn't show here, you would see how it is spreading as well. So this is definitely a problem in Alberta. Now, chronic wasting disease is an infectious disease. It is actually the most infectious prion disease that we know. It is taken up through either through the skin or nasal mucosa or ingestion into cervix. Wild-tailed deer, mule deer, moose, elk, and caribou can have it as well. We have not seen that one in North America yet, but in Europe. Once the disease has been taken up, there's an infection event and in what we call neuroinvasion, meaning from the digestive system, the prions migrate all the way up to the brain. When they reach the brain, the disease becomes symptomatic. This period can be in the range of 2 to 5 years, which is in the incubation period where the animals are still look perfectly healthy, but in the inside they're already infected and it's getting worse. Once the disease reaches the brain, it goes relatively rapidly and the animals get really sick and decline and die. During the time, even while they're still healthy, the animals are shedding infectious prions through their feces, urine, milk, saliva, placenta, nasal secretions, and in the end through the carcasses as well. Prions, unfortunately, are very resistant and they remain in the environment, so that closes the circle again, so that we then have another uptake event and the infection spreads that way. As you can see on the right-hand side, we do not know if chronic wasting disease has the ability to transmit it to humans, which is particularly, of course, a point of concern for hunters and indigenous people who rely on cervix as a source of protein. Another possibility, which we have seen with prion diseases, that they can jump from one species to another, not only from the cervix to humans, but potentially also to other agriculturally important species. And if chronic wasting disease would jump into them, it would be a major situation there. Now, how can we try to prevent uptake and disease spread? This is where vaccines come in. We envision that a vaccine would have the ability to potentially stop the uptake and the first step of the infection. But even when inside, it may then stop the spreading throughout the animal, the neuroinvasion. This is potentially a target of the vaccine, but also the shedding. If a vaccine can prevent the shedding of the prions, even if an individual animal may still come down with this, but if it reduces the shedding quite a bit, this would be a major target of the vaccine as well. So we have different points where we're interested in see if vaccines are efficacious in preventing uptake and neuroinvasion and spreading and the shedding of the prions. Now, my lab was far from the first ones who start on developing prion vaccines, and there were actually many more labs, but now there's only 3 of them remaining in Canada. The 2 labs that started earlier were the laboratory of Scott Napper at the University of Saskatchewan in Vido. He developed a system of disease-specific epitopes, as he sees it, as a vaccine, and he's still working on testing this one. Another lab is the laboratory of Hermann Schatzel at the University of Calgary, and they're expressing recombinant versions of the antigen, meaning recombinant version of the prion proteins, in this case as a dimer, and producing nanoparticles as their vaccines. And so these are basically my competitors, the competitive labs. About 10 years ago, we joined the fray and started my own research in the structure-based vaccines, looking at exactly the structure biology results. And so in the end, it was the 3 labs who were competing. However, we decided to actually pull together. So we have several projects where we're collaborating, we have grants together. And our theme is that Mother Nature decide which of these vaccines is best and hope that at least one of them will prevent disease or help us bring it down. Now, structure biology. The infectious prion protein is, of course, relatively difficult to work with because on one side it is infectious. On the other side, it starts to clump into protein clumps. And here you see some electron micrographs, meaning very high power magnification images of these, what we call amyloid fibrils, that is full of the infectious form of the prion protein. With the advent of modern cryo-electron microscopy, it has become possible to study them at much higher resolution. As you can see in here, some of these cryo-electron micrographs were used to process these data. And here you see a highly processed fibril there. And if you look closely, you see these striations. And each of these striations represents a single molecule in these fibrils. If you take a cross-section across this fibril, this is what you see here. And what looks a little bit like a Christmas tree here and there, this is literally the individual amino acids that make up the protein. And here on the right-hand side, you see it higher magnification. And from density maps, as we call them, like these ones, we can actually figure out where each amino acid sits in a structure like this one. So this was a major breakthrough in structural biology of the prion protein. By now we have 7 different structures from different types of prions, meaning different prion strains that have different properties, can infect different species. So some of these ones are sheep-scrapie prions. There's a human version of prions, another Gerstmann-Sträussler-Scheinker variant, which is a little bit different from what we were talking about. And then there's, of course, chronic wasting disease prions that I had on my very first slide, and you see them down here. So we have detailed knowledge of the structure of the prion that was actually isolated from a white-tailed deer that died from chronic wasting disease. So we have detailed insights into these structures. However, when we thought about putting together a vaccine proposal based on structures, this predates these structures by quite a few years. So this crude drawing here on the left-hand side is basically a model that we were basing our idea of the structure-based vaccines or model-based vaccines, to be more precise, on this one. And you see it is very different because an individual molecule of the prion protein is represented by 4 layers, 4 rungs as we call them, sitting on top of each other. This crude drawing was supported by a whole range of indirect data on various forms. And when we had these data come out, I thought we could take advantage of this one and we can predict which of these amino acids would be sitting on the surface of the infectious prion protein and potentially become a target for a vaccine. So I asked my student, Andrew Fang, to select some of these options. And for example, he selected this option. So he elected these 7 amino acids highlighted in green and cyan and thought they may come together on the surface of the prion protein to form what we call an epitope, meaning something that is recognizable only in the diseased form of the prion protein. If we look in the linear sequence, or just a lining up of the different amino acids, you see that these 7 amino acids, again highlighted in green and cyan, don't sit together in the linear sequence. And if we look into the native structure of the prion protein, and this is a structure that was solved using X-ray crystallography here at the University of Alberta quite a few years ago. The same 7 amino acids are again highlighted in green and cyan, and you see in this structure, which is a healthy form of the prion protein, these 7 amino acids are spread over the surface and don't come together in an epitope. This is important. In our prediction, you saw the crude drawing earlier on. We had predicted that these 7 amino acids would come together on the surface. This is a more recent model that came after we had started with the vaccine project, and the people who put this model together had equally predicted these 7 amino acids to come together. So I can use this illustration to show you what we think the structure of the infectious prion protein would look like. And you may notice it looks different from what the cryo-electron microscopy data shows these days. However, this was the basis of a vaccine, and I will show you why that is important in a moment. We took these 7 amino acids and transplanted only these 7 amino acids into a totally different protein. And we used the fungal het S prion protein, the one that I had mentioned earlier, and inserted this one into the scaffold protein to make this one the carrier for our vaccine. Now, the het S protein is very useful because we can study it in great detail. And it has nothing to do with infectivity. It's not a pathogen at all. So it has a great safety profile, which is, of course, important for a vaccine. Now, the HET-S prion protein had been studied using a technology called solid-state NMR spectroscopy, and the people who published this data showed that this protein can form amyloid fibrils, somewhat similar to the ones that we have seen in prion diseases as well. There are some differences though, but still somewhat similar. We know that this protein is easy to work with. For example, you can see it here. It is not disease related. It's actually determining the mating type for this filamentous fungus. So when you grow them on an agar plate, if they're compatible, you see they can fuse and form spores. And if they're incompatible, these barrage lines are formed, meaning the cells are killed off because these types of the fungus are incompatible. And the HET-S prion protein plays a role in this one. That's its function. So it's a very safe protein. We know what it does. It is very harmless. And we have a great structure to work with. So what we have done when we designed our prion vaccine, we've taken the same 7 amino acids that I've highlighted earlier that Andrew had selected and that are shown here in cyan and in green. And as you see, we inserted them in what we call a discontinuous structure-based manner. Discontinuous meaning they're not together in one linear sequence. We spread them out, and structure-based, we selected them to sit just on the surface of this HET-S protein, scaffold protein, just on this side of the surface here, to be exposed this way so that the immune system could see this one, which is why this one would be a vaccine. That's what was our thought behind it. Now, this protein has an advantage. When you express it in this way, we can make it adopt this structure, and when it does, it forms fibrils again that we can see in electron micrograph, which you see here. So this is our quality control. If it forms the fibrils, we know it adopted the right structure. However, to be certain that it really took on the right structure as we had designed it, we went back to the people who had done the solid-state NMR structure, just a couple of slides, this past slide. They used the same technique, but now used both the HET-S that had been used as a basis, and then used our vaccine. And you see that most of the data are identical. Few amino acids that we had changed sit in different positions, proving to us that actually the vaccine adopts exactly the right structure as we had predicted, which is of course important for understanding how our vaccine works and when we want to make improvements and more changes about it. Now, when we can make this vaccine, it is very easy to produce. That's the advantage of using the system. We have the gene for it. We can insert it into bacteria, grow the bacteria in the lab, harvest the cells, lyse the cells, pull the protein out through various steps, purify the protein, and then refold it in the test tube into the right conformation. And this is then the pure protein in the right conformation. That is our vaccine. In order to make it the vaccine, we break the fibrils into shorter bits and pieces and add an immune stimulant called an adjuvant to make an even stronger immune response in the animals, and that is our vaccine. So the advantage of this one is that we can produce it in significant quantities, and it's pretty straightforward. This is standard technology in a biochemistry laboratory, and it can be scaled up if necessary. So if we are successful, we can definitely produce significant quantities of it. Now, the next question we had is, of course, so we engineered this epitope based on our prediction, a relatively crude prediction in that one. Now, does it give a good immune response? This is what we did there. We took some wild-type mice, and we injected them with the vaccine and the adjuvant. And we used the prime dose, and we used 3 boosters because we did not know how strong the antigen would be. We took pre-immune sera to see what the immune response was before it was, and post-immune, and steps in between, so collecting blood and making serum out of it. And then we wanted to test this serum. Can it recognize prions? Because if our vaccine copies a little bit of the infectious prion protein, this antiserum from these mice should be able to recognize prions. And that's what we did. We took the blood and we tested it against prion samples, in this case chronic wasting disease infected brain homogenates. And as you can see, this black bar means that our antiserum from mice that were immunized with our vaccine does indeed recognize chronic wasting disease prions. When you use uninfected brain homogeneous control, there's no signal. And we also immunized some mice just with the scaffold protein without those changes, the het S by itself. And the antiserum from these mice is unspecific, meaning it cannot recognize the infectious prion protein. So this was a great success. We knew now what we had engineered there. It worked. It allowed us to recognize the infectious prion protein. And yes, this was not the only one. I'm only showing the one candidate that produced positive results. Andrew had produced several other vaccine candidates that failed at this stage. The immune response in the mice was not able to recognize the infectious prion protein, which is why we didn't study those any further. Now, I just want to explain to you the competitive ELISA a little bit that I just showed you on the previous slide, because it's an important tool and it comes up a few more times. So in a competitive ELISA, we take the antiserum from an immunized mouse and we mix it with a sample, in this case, for example, a brain homogenate. And if the brain homogenate comes from a healthy animal or a healthy person when you use human samples, then these antibodies from these immunized mice would not recognize the prion protein here because it's a healthy form of the prion protein. Now in our test plate, our ELISA plate, we had coated the plate at the bottom with our antigen, with our vaccine. And so the antibodies, since they were raised based on this vaccine, are very good at recognizing the antigen at the bottom. So you see all the antibodies, or almost all the antibodies goes at the bottom. We have a detection system which involves another antibody and an enzyme reaction, and we get a strong signal. That's a positive signal, meaning these antibodies recognize the plate. Now in the competition ELISA, when we now use a brain homogenate from an infected animal or an infected person with a prion disease, and we have prions in there, and now these antibodies would recognize these prions. This complex, now these antibodies are taken care of. They are no longer available to bind to the bottom of the plate, so you get a much reduced signal at the bottom of the plate. And if you take the readout, you see the reduced one. And what I'm going to show you in many graphs is what we call a delta OD, so difference between one from the other value. So when you see this one going as a delta OD, that is where it's coming from the competition ELISA. Now, we had this vaccine. We saw with the competition ELISA that it was capable of recognizing infectious prions. Now we wanted to test it in an easy-to-handle system. This is where this rare genetic prion disease in humans comes in. I'm just showing this one. This is data from humans. So people who carry point mutations, meaning single amino acid changes in the prion protein in their brain. And there's 3 different forms that I'm showing here. We know quite a few more than that, 3 different mutations. They give you 3 different forms of the disease. And in all of these 3 cases, if you're a carrier of the mutation and you live to an age of 50 or 60, your chances of developing disease is essentially 100%. If you live long enough, you will come down with the disease, including this rare genetic disease, Gerstmann-Sträussler-Scheinker disease. Now, this is of interest to us because we were able to obtain a transgenic mouse model that carries this very same mutation. In this case, in humans, it is the amino acid protein is replaced with a leucine at the codon 102. And if you carry this one and you live long enough, you will come down with the disease. And it's an invariably fatal disease in humans. In this mouse, the numbering is 1 off because the mouse protein is a tiny little bit shorter. So it's P101L in mice. These mice that were provided to us by Glenn Telling from Colorado State University overexpressed a mutated form of the prion protein. It has the consequences that these mice by themselves start to come down with the disease at around 177 days of age. And by 200 days, you see all the animals are diseased, and disease is rapidly progressing that by 220 days, the animals are so far advanced that we have to euthanize them for animal health reasons. So you see the disease is very rapid once it is onset and is invariably fatal to these animals. This is our mouse model. Now, this is the same curve I just showed you. This is in unimmunized mice, and we have done this many times over. If we take these mice and we immunize them with this het S scaffold protein, but without any of the changes, as you see, these mice don't gain any benefit from the immunization. strictly speaking, they even get the disease a little bit faster, although we don't know if that has any significant meaning. If we give these mice our vaccine, in this case without any adjuvant and without any immune stimulant, you can already see these mice stay healthy. This is the health curve, much longer already. If we use immune stimulants, as in these adjuvants, you see we can keep these animals healthy much, much longer. So we have an extension of the health span of more than 280% in the best of cases, as you see here. However, in the end, the disease breaks through, the animals do die of the disease, and it's a prion disease, they come down, but after a much prolonged time. Now, this little bit crude video shows you these animals at about 200 days of age. These are unimmunized animals, and as you can see, they are in a poor state. These animals are at the end stage, meaning they can no longer reach the food and water, and by our animal protocol, we were forced to euthanize them on that very same day. So this is a mouse in a poor state. Now, in the next moment, this video will switch over to age-matched controls, meaning these animals have the same mutation, but they were the ones that received the vaccine. And they're 200 days of age. This is what a healthy mouse should look like. So they have no disease symptoms whatsoever, and this stays on this for these mice until about 500 days. They come down. So we were really happy with this result. And yes, we did a lot of controls because in the beginning, I did not believe that we did everything right to make sure that these mice really do have the mutation, the ones that you show in a moment there, that were immunized. And yes, they do carry the mutation. So we are certain that these are the same mice have the same mutation, only these ones were not immunized and these ones were the immunized ones. So they got lucky on that end. Now, I had shown these data at a seminar at some point, and Samantha Allen and Peach Van Wyk from the Wyoming Game and Fish Department, who work also at the Thorne Williams Wildlife Research Center in Wyoming, contacted me and told me that they are working on this research facility under the auspices of Wyoming Game and Fish, and that they have elk, Rocky Mountain elk on their premises on the research center there that were not in the experiment at the time. They had been on the premises for a long time, already for over a year already, meaning these elk have been contaminated, presumably infected with chronic wasting disease already. So it was not a real efficacy trial. However, they allowed us to send them some vaccine and they would immunize these elk, which we of course happily agreed to. So we were able to use this elk trial in Rocky Mountain elk to test the antigenicity and the immune response against our vaccine in elk, which of course is an animal species that our animal facility here at the University of Alberta cannot handle. We can only handle mice and hamsters, small rodents like this, not 400-pound Rocky Mountain elk. Now we had 3 groups with 4 animals each. One group got only a buffer controls, just as a negative control. Then another group got 100 micrograms of our vaccine, and the other one got 200 micrograms of the vaccine injected into the muscle, same as we usually get our vaccines together with an adjuvant. And here you see again the prime dose and the boosting scheme, and also when blood was drawn in these animals. So this was our experimental paradigm here. These were pictures sent to me by Peach Van Wyk and Samantha Allen from the procedures that happened. So here on the left-hand side, you see the vial of the vaccine that we had shipped out to Wyoming. And here you see them working on one of the elk cows, immunizing them and collecting samples for us that we could analyze. So this is our vaccine in a large-scale animal model here. And when we got the samples, we obviously wanted to see, do we see an immune response against our vaccine? We're hoping to do so because we saw strong immune responses in mice earlier, and in elk it is the same thing. The 4 animals that only received the buffer, of course, did not produce an immune response. You see the bars are the pre-immune and the post-immune bars are both low. The animals that received 100 micrograms showed a nice strong immune response, and at 200 micrograms it was even a little bit stronger. Now this is the typical antibodies which got IgG, standard antibodies that are found in the blood, so that we saw in the serum. We also, more recently, we checked the serum if a different form of antibody was raised, and we did not expect that to happen. When you give a vaccine in intramuscularly, you usually get an IgG response that we see here. IgA is a special class of antibody that protects the mucosal membranes. And in an intramuscular immunization, you do not expect to see an IgG response. And as you can see here, none of the elk showed an IgG response. This is exactly as we would expect it. Now, why is that important? IgA is protecting the mucosa, and since chronic wasting disease is, of course, taken up orally or nasally through the periphery, if you want to have a vaccine that protects against these diseases, you ought to have an IgA response. And the intramuscular immunization is clearly not the best way to go about this one. We knew this one ahead of time. This study was really at this stage, seeing the immune response in elk for the first time. We also used the hunter sera that we had received from these elk to see, and again, a competition ELISA, can they recognize chronic wasting disease prions in one of these ELISAs? And wherever you see a blue arrow here, these are the antibody responses that were positive, meaning these animals, 3/4 of the animals that were immunized with our vaccine had now immune response in their blood that was capable of binding infectious prions. 1/4 was not positive, but this is a range of what you see with every vaccination. So this is not a great detriment. Now, pretty much at the same time, we also started to work with the Mathiasen lab at Colorado State University, where there's the Prion Research Center in Fort Collins, Colorado. And they have a very unique facility because they have a facility that can house white-tailed deer indoors in a biosafety level 2 animal facility that allows them to infect these animals under controlled conditions with chronic wasting disease prions. This is a very unique and rare facilities, and obviously, as you can imagine, housing animals under these facilities and working with them under biosafety level 2 facility, as you see the PPE that you have to wear is extensive. This is an expensive operation to run, and we collaborate with them to test our prion vaccine in white-tailed deer under these controlled conditions. This is a project that is NIH funded. Of course, the lead is by Candace Mathiasen at Colorado State University. Here we're collaborating with Hermann Schatzer's laboratory out of the University of Calgary with their PAPC-directed vaccine. I don't want to go into great detail there, and our vaccine. So this is a collaboration from the 3 laboratories. Here you see the immunization and blood draw schedule for these white-tailed deer. So it's very similar. The dates are a little bit different, but the rest is very similar. We have a prime dose and we have boosters, we have blood draws, so we get some samples that we can analyze. And we had 1 animal that received no vaccine, 3 animals that received Dr. Schatzer's PAPC vaccine, 3 animals that received our vaccine, and 4 animals that received both vaccines. Again, this one was intramuscularly given with the same adjuvant. However, this was just an immune response trial. These animals were not infected. We just wanted to see the immune response that developed after the immunization and also the safety profile of the vaccines. And I'm happy to tell you that we saw no detrimental effects on these animals, meaning they took the vaccines just fine. There was no side effect, which is good because otherwise we would have a problem on that end. When we received these samples, obviously we ran the same ELISAs again, just checking again of the immune response. And as you see, all the vaccines that received all the deer that received our vaccine developed a strong immune response, shown by the green bars. Even the animals that received both vaccines also had a nice, strong immune response against our antigen, as we would expect it. The green bars are after 15 weeks. However, after 1 year, this is the blue bars, you see the immune response had declined quite considerably. However, when these animals were given another booster immunization at the 52-week mark, just 4 weeks later, you see the immune response is right back up, which is a good sign, because that means the animals had memory cells in their body, which were then stimulated and responded right strongly against by producing more antibodies that we can measure then at the next sampling interval. And this was true for both the animals that received only our vaccine or both vaccines together. Now, interestingly, when we tested the immune response for the ability to recognize chronic wasting disease prions in this competition ELISA format, we saw that 2 out of 3 animals that had received our vaccine, shown here, were capable of recognizing chronic wasting disease prions, same as we had seen earlier in the elk. However, the animals that received both vaccines did not. And this is still a puzzle that we had not solved yet, why that is, why there seems to be some interference. It is definitely something to think about and keep an eye on. We again took the antiserum from these animals and tested it for the presence of IgA antibodies, and as expected, there was essentially no response to it. Again, this is as expected. Now, in a second trial, in the same project, we now wanted to see the efficacy of our vaccines. Again, the Schätzl vaccine and my lab's vaccine. In this case, we were able to get 9 deer in the controlled facility. 3 deer were given no vaccine, 3 deer were given just a PPC vaccine from Dr. Schätzl's lab, and 3 deer received our vaccine. Again, in our case, it was given intramuscularly with the adjuvant. What was different now is that after the animals were immunized, they were given 300 nanogram of brain homogenate from a chronic wasting disease infected deer. This is the point of infection here. So this is an efficacy trial. Can any of our vaccines prevent infection or delay infection, in this case, under these very controlled conditions? We received the antiserum from these animals, and as we fully expected, the animals that had received our vaccine produced a nice, strong immune response against our vaccine. And the ones that did not receive our vaccine, either no vaccine or only the Schätzl vaccine, could not recognize our vaccine. Again, everything exactly as planned. We did, again, this competition ELISA to see which one of these animals could recognize chronic wasting disease prions. And again, 2 out of 3 deer that received our vaccines could recognize it just nicely. Same numbers as before, so this seems to be a repeating pattern. Now, where are we with these challenged animals? So at the moment, all animals are still healthy. These animals are tested regularly. We can do antemortem testing, meaning testing while they're still alive before they show symptoms, by collecting lymph nodes from the rectum, because they're easily accessible there. The lymph nodes, the rectal lymph nodes of cervix of deer, are known to be showing prion infection early on, quite a while before the disease manifests in the brain and becomes symptomatic in the animal, which is why these animals regularly have rectal lymph nodes taken. It is a little bit invasive, which is why it cannot be repeated too many times, but it is an early detection sign for us to see if the disease is developing or not. In one of the sampling, we had 2 out of 3 of the deer that received no vaccines had already positive lymph nodes at 15 months. However, to receive a proper prion diagnosis, it needs to be positive again when they're checked 3 months later. And when they were checked 3 months later, they were negative. You have to understand that obviously the lymph nodes that were taken at 15 months are removed, and at 18 months, a different set of lymph nodes is taken, and these ones were still negative. So strictly speaking, all animals are still negative. So this graph is still true. We consider all animals at the moment show no clear sign of infection. And this is where the efficacy trial is at the moment. So this is the status where we're at. Obviously, as you can imagine, we are very impatient to see what's going to happen there. But this one will come down in the next year. We'll see the data there to see if any of our vaccines do any good to these animals. Now, I had mentioned that we wanted to also reformulate our vaccine so that it could be taken up through the mucosa. Now, when you take a normal vaccine, protein-based vaccine, as we had developed it, and you would administer it to a mucosa, you would have either no or very, very limited uptake, meaning it would not be an efficacious vaccine whatsoever. However, in a paper from a few years ago, I read that by attaching what we call an amphiphyll, which is a structure that you see here, covalently attaching it to the protein, you can make it a mucosal vaccine. By attaching this amphiphyll, this one now binds to a particular protein in the mucosa, the serum albumin, in the mucosa of this deer. This one was an invention by Brittany Hartwell and was published in 22, and she did this one on a different antigen in her system and tested the mucosal immune response in different samples from mice in this case. And you see that the blue signals clearly show a nice strong mucosal immune response, while when there was no attachment of this amphiphyll, there was essentially no mucosal immune response or very, very limited. So, so this looked very promising. So this is what we wanted to try. Now, in order to attach this amphiphyll covalently, there are some biochemical methods that we can use. Here, this is what we call a gel electrophoresis. So this is where we purify samples and we can run them by size. And see, this blue band here is the monomer single molecule of our vaccine, a whole bunch of them running in here. So we see the size. And when we attach the amphiphyll covalently, you see the size changes. It runs at a higher, it runs at a slower speed and higher in the gel. So it showed us that we successfully attached this amphiphyll to our protein. So this looked good. Here on the right-hand side, you see another electron micrograph of this very same samples, and you can see they still form fibrils. As I mentioned earlier, the ability to form fibrils is our quality control measure for this vaccine. By seeing these fibrils, we could say positively that these samples still folded in the right way, meaning that these amino acids are still exposed in the same way as we had seen it before. So this is a very important quality control step for us. Looks very similar. We have, and this is ongoing, this is a relatively new, it was only started earlier this year, another efficacy trial in Rocky Mountain elk. This is a collaboration now with the Napa lab, Scott Nappa's lab from Vido and University of Saskatchewan with his vaccine, Herman Schatzel's lab with his PAPC vaccine, and my lab with our vaccine, and a collaboration with Peach Van Wyk and Samantha Allen and Charlie Banson from Wyoming Game and Fish. So we use it, we are back at their facility. They were able to provide us with a new batch of yearling Rocky Mountain elk cows that were then immunized. In our case, immunized mucosally. For the other ones, they used other procedures. And as you see, we recently had another booster for these animals, and we're expecting these samples to come in at some point in the future. So this is still ongoing, and we have a limited set of data yet. So we had, in our case, we had some control animals that only received the Het S variant, the mucosal Het S variant, and then the mucosal vaccine.
Now, Dr. Wille, sorry, yeah. Dr. Wille, it's Andrew here. If you were able to wrap up in the next couple of minutes or so, that would be great.
Yeah, I'm almost finished.
Excellent, thank you.
These ones were given into these animals. The animals were all negative with Ramalt. As you can see here, this is the atomizer that sprays the vaccine up the nose of these animals. You see this one is being applied to the elk nostril of the elk. It is sprayed up the nostril, and you see the elk responded quite forcefully there. We again had a nice serum immune response in these animals. We tested this in the serum. Now we see an IgA response in the serum, so this looks good. And we now also tested saliva of these animals, and we see the IgG immune antibody in the saliva, and we also see the IgA in the saliva, so the potentially protective antibody comes in a special form in the saliva. So this is where we are with these ones. We do not know if these protect these animals. This has to be shown in the future, but this is where we are in the trial. To sum up, so we developed the vaccine, our prion vaccine. It works very well in these transgenic mice that I just described. Produces strong immune responses in white-tailed deer and Rocky Mountain elk. And the trials are still ongoing. They take quite a while. We just started the mucosal vaccine trial, and we see an IgG immune response in the elk serum and saliva. And so we're awaiting the outcome there. We do want to explore additional protein scaffolds to extend the immunity, what we have seen there, to have additional epitopes, but that's work that's going into the future there. We want to test the mucosal adjuvants, which we had not included in the earlier trials. And we are just looking together with Scott Napper, he's the lead scientist on this one, to do an efficacy trial in Rocky Mountain elk, presumably in Saskatchewan. Details still have to be figured out. In the end, of course, I have to thank the people in the lab who did the work, the collaborators highlighted in red, and of course, I have to give a big thank you to the funding agencies without whose support this work would not have been possible. And I'd be happy to take any questions.
Wonderful. Thanks very much, Dr. Wille. What a great presentation, and I would say exciting and encouraging area of research. I want to thank everyone for joining the science seminar today. Please stay tuned to learn about the winter seminar series, which is currently lining up. The first seminar will be on Tuesday, January 27th in the new year, so you'll see an announcement for that. For those that are not on our mailing list and would like to be added, please contact the Office of the Chief Scientist. That's at epa.ocs@gov.ab.ca. And with that, we'll end our presentation for today. And thank you very much again, Dr. Wille, for a really exciting and interesting presentation. Thank you, everyone.